First off, no one in America can say my last name properly, so thank you. So, as mentioned, I am our head of patient advocacy at Taysha Gene Therapies. I have been with Taysha for about 2.5 years, and in that entire time I've worked on the Rett Syndrome program. So it's great to be back in the UK, my first time in Manchester, but I may have met some of you at one of the galas a few years ago, and thank you again for bringing us together today. So before I start, as you kind of heard in the previous presentation, it's important to remember that what we're talking about today is an investigational gene therapy product. So this product has not been approved by any regulators, including the MHRA in the UK, nor is there any guarantee that it may be in the future. So we'll just keep that in mind that we're running clinical trials to determine if there is a potential approval in the future. So to that point, there's a lot of things that we may not be able to answer for you today, but we will do our best to answer those questions that you do have. And I'll be around all day and all afternoon, so please feel free to come up and say hello. So I have just a few slides here today, and then I'm going to hand it over to Dr. Dan Lumsden, who, as you I think have collected by now, is our principal investigator in the UK for our clinical trial site here. But again, as you all should know by now, Taysha is investigating a potential gene therapy for Rett syndrome. And once again, thank you, Reverse Rett, for having us today. So in my role of patient advocacy at Taysha, I am really responsible for ensuring that our company is understanding the perspectives, the insights of families, specifically in the UK, US, and Canada, where we're running trials, and keeping those best interests and the wants and needs of these families at heart, and incorporating that into every decision that we make at our company. So not only do I partner with the patient advocacy groups, but I am advocating for you all internally. Rachel's the first to tell me this is something that the UK community wants, and I relay that back to my company to make sure that we're keeping those things top of mind. So these are just some examples of how I kind of navigate my role at Taysha. As I mentioned, understanding the unique lived experience of the folks that we're working with, and then really, as Dr. Lumsden mentioned, translating what you all would want to see benefit into what is something that's measurable in a clinical trial and is not causing too much burden for families who are participating in the trial. Because ideally, we would have a list of 10 to 12 things that we would be measuring, but that would be an extravagant amount of work for us to collect in a clinical trial. So there's this nuance to understanding what are the most important aspects that you would want to see change in, and how do we translate those into an endpoint in the trial. We also work to determine from families what is the information that you all need. So the more questions we get, the better it is for us to learn what areas are you looking to learn more about. And we can work in the future to develop some educational information to cover those. So the larger portion of the presentation that Dr. Lumsden is going to walk through today is our attempt to answer a lot of the questions that we've received in the past around gene therapy in general and clinical trials specific to gene therapy. And then the last mention here is just kind of the co-creation of the clinical trial experience. So for those who do choose to and are accepted into a clinical trial, we really want to make sure that we are reducing the burden on families as much as possible. As you'll gather from later slides in the presentation, it's a huge commitment to participate in this trial. And we really want to make sure that folks understand that before they sign up and are carried through the process appropriately so that they feel confident with whatever decision they make. So here's just an example of some of the ways that we've partnered with patient advocacy groups such as Reverse Rett and others in the past, and gathered community insights directly from US, UK, Canada, and Israel. So patient advocacy partnerships are essential to what we do. Rachel, Andy, the whole team have such a great pulse on what's going on in the UK, and they share with us their sentiments. We have ongoing interactions where we're talking about a variety of topics, but to date, the patient advocacy groups have reviewed our protocols and advised us on our study design, what endpoints we're selecting. They help us to identify where would a potential clinical trial site be. Again, how can we best support families with travel when we're looking to provide that assistance in a trial? And then last but not least, in our regulatory interactions. So I know most recently Rachel and the team had the ability to gather some videos from some folks, maybe some of you are here, but they were able to create a really nice video that was incorporated into our submission to the MHRA, which just really helps us to share the patient perspective, the caregiver perspective with those key audiences. And then it's a few years ago now, but just want to once again thank the families that participated in a survey that we did back in 2021 when Taysha was first kind of getting into the Rett syndrome space. That really helped to form the clinical trial and the early work that led to the clinical trials that are starting these days. So thank you, and stay tuned because there will be more surveys and opportunities in the future. But again, feel free to come up to me, ask questions, tell me your opinion. The more I hear from you, the better. And then before I hand it over to Dr. Lumsden, I just want to reiterate our commitment to this community. We understand that time is important and that we all want the best thing for this community, and our team works with that urgency in mind. We have a team that is working as quickly and efficiently as we can, but it is important to remember that drug development does take time. And Dr. Lumsden will speak to some of the stepwise approaches and why things seem to take a little bit longer than we all would want to, but there's reasons for that, and hopefully you all can understand that after seeing some of this information. We as a company strive to provide timely, accurate, transparent updates as best we can. We do have to abide by local regulations, specifically in the UK, so with that said, there will be things that we are not able to answer for you today, but thank you for understanding that, and please don't hesitate to come say hello later. I'll hand it over to Dr. Lumsden.
I'm really sorry, I'm back up again boring everyone, so I do apologise for a double show as it were there. Just to pick up on one of Chelsea's point there, the patient advocacy group, of the work that is done by all of you and spearheaded by Rachel, Andy and the team, it's absolutely essential to moving any of this forward. You do not get near the MHRA letting you do trials, getting any kind of approvals, any kind of commissioning. So what you are all doing in this room is hugely and vastly important for the work there. What I'm going to concentrate on is a little bit of a reiteration of some of the technical sides, but also then talking about some of the practicalities of the trial side. So we know we've talked about this a lot. We're talking about gene therapy and to provide an overview. So when we're thinking about gene therapy, we're talking about monogenetic diseases, so diseases that are caused by a change in a single gene there. The goal of the gene therapies then, as we've discussed, is to deliver a healthy and functioning copy of that gene to the cells in the body that it needs to get to. Be that the liver in some cases, be it the brain and the spine in this situation there. Lots of different ways in which we can get the gene into the neurons of the brain, but it's viral vectors which both of the trials and importantly the Taysha study is using there. And the AAV9 has been around for about 50 years of experience using it there. It's essentially the most studied platform for delivery of the gene therapy. Really importantly, gene therapies are typically designed to be administered as a one-off treatment. The idea is you get one shot to give it. It's not a therapy that you need to top up and give repeated doses there. And as we discussed before, you're trying to get to the root cause of the problem and correct it there. So for most people, not all, but most people, Rett syndrome is caused by a mutation or some abnormality in the MECP2 gene, and that gene change then leads to a lack of the MECP2 protein there. When we have too little of that gene there, we end up in the situation which gives rise to that clinical picture of the Rett syndrome there. The problem is, as we've said, if we express too much of this, we get that Goldilocks principle going the other way, and we end up with a duplication syndrome there, and we end up with a clinical phenotype caused by that. So the balancing act here is not just to get it in, it's to get it in and express it at the correct level there. So just consider briefly then some important facts about gene therapy and really thinking about how these kind of trials are different to other medication trials that you might have been aware of. So there's lots of ways in which they're different there, really. And I think it's important that this really figures into and factors into the trial design, the follow-up of young people, and some of the practicalities of how it might be commissioned moving forward there. So gene therapy, it's a one-time treatment. So you get the dose, you get the vector delivered by that virus on one occasion. And the problem with that is because it's a virus, the body then can potentially demonstrate or develop antibodies. That means you can't get another dose. So it's a one and done. That means that if you're taking part of any of these trials there, you won't be able then to take part in a study where you subsequently get a second dose or a top-up. and that's an important thing for us to consider there. We've talked about it, but there is that risk of an immune response to the virus. Amy's going to expand upon that later in the talk, but a lot of the protocol is designed to monitor for this. It's safety. This is why there's lots of blood tests. This is why there's lots of imaging of nerve conduction, which is a necessary evil. The burden is there, but the burden is there as a necessity. And I think that's really important that anybody considering taking part of these trials is aware of that. So what are the goals of a phase 1-2 trial? Well, we've discussed this already. This will not tell us how well the gene therapy works. This is going to establish that it's safe, that there's signs that there is some efficacy, and then we'll move forward into bigger studies that will more definitively answer those kind of questions there. So these phases are a really important way to make sure you don't expose large groups of young people or adults to a therapy that then subsequently it isn't safe. And there's a whole monitoring and process with the regulators and within the internal trial design to ensure that that safety is put first. First-in-human trials are really put under an appropriately high scrutiny, and certainly any of us, it's even within our institution, the amount of appropriate sort of committees and boards and monitoring that's in place there. Please be reassured that this is not anything that anybody would take lightly there. Because these are phase 1 studies, the eligibility criteria have to be quite restrictive. The eligibility criteria, the list of characteristics of people who get into the study. It's important that then that gives us a sample that's representative of the group as a whole. But if we have too broad an inclusion criteria, it can be difficult to pick up on those safety signals, and it might be difficult to pick up on the efficacy that's emerging from that there. They have been deliberately designed to bypass those kind of problems there, and of course the hope is that with subsequent trials and in the future, it'll be able to increase the inclusion criteria, but we're limited to what we have now for very good reasons. And I think it's a very appropriate sort of inclusion criteria that have been chosen. So in terms of the monitoring of this safety, this is something which is taken very, very seriously. And I think it is important for people to be aware of there. There are differences because it's first in human, it's a phase 1 and 2 trial there, and a really important part of this is the Independent Data Monitoring Committee, the IDMC. It's something you'll see over all of the literature and any of the patient information sheets. This is essentially an independent group of clinicians and scientists who've got experience in trial methodology, have got experience in the genetics and the molecular biology behind it, and have got the skill set to be able to negotiate decisions around the safety of the trial dosing. Things go ahead in a very stepwise approach, and it's really clear that it's the early data from each of the first few participants that's put through the trial that will then allow that committee to make the decision to step forward with the trial. But this is why it feels very slow, the initial recruitment. It's because there's a very high and appropriately high level of scrutiny to make sure that there's no suggestion that it's not safe to move forward. And be reassured this is things that people take incredibly seriously and appropriately so. Okay. So who carries out the clinical research? There's lots of different people that are involved in it. So the study sponsor is Taysha. So essentially the sponsor has the responsibility for the overall trial running. They have to select appropriate investigators. So I had to prove to them that I could do the job. Hopefully I did all right there. But they also have to then identify sites that are going to be able to deliver the trial safely, have the right patients coming through those services there, be able to do all the appropriate monitoring and make sure there's no problems with that there. The sponsor basically holds the risk, and they're responsible. That's that legal responsibility there. The regulatory bodies, these differ in each country. So the FDA is the Food and Drug Agency there. The MHRA is the Medicines Health Regulatory Agency, and that's the body that's approved the Taysha trial in the UK. The next level of that is NICE and the commissioning bit of work there, but that's a discussion I think for a different day there. On the clinical trial sites, there is a lot of infrastructure that's in place to ensure the safety of the conduct of the trials, and really it's the role of the principal investigator on each site to ensure the safe running and conduct of the study on that site there. They're responsible for all the training of the team, the completing of the protocol, and ensuring that all of the appropriate assessments are done in a timely fashion there, and they're also responsible for the safety of their participants. So it's the decision of the principal investigator who is included in the trial ultimately, and if there's a need for someone to come off the trial because of a safety issue, then that will be down to the principal investigator. Of course, for a gene therapy study, the problem is we can't take the gene therapy away. Yep. Once it's there, so it's different in that sense to other trials there. Really importantly, I think, The sponsor is blinded to any of the data that comes back. So we will know, and any other site that runs the study will know which children and where the data comes from, but those identifiable information is not passed on to the sponsor there. So they're blinded to that information. So there's lots of different steps to the trial participation. When this opens, this is what people go through. The first part is the screening period, and this is a period of assessment to make sure that young people meet the inclusion criteria for the study, and there's no exclusion criteria that would mean that they're not suitable to continue, or any safety issues that would preclude them from being involved. Usually that involves a number of different visits to the sites. There's a lot of different investigations that will be part of that. But be assured, again, this is all around ensuring that it's safe for each individual person to move forward with the trial there. The baseline visit's really that starting point of, this is the data that we're going to compare our outcomes to. It's the point where you've been included in the trial, there's no reason to be excluded, and that's your data capture that you're then going to compare for the improvement. For some studies, there's a degree of randomization. So in other trial designs, you might be randomized to placebo or a therapy. For these studies, for the Taysha study, there is not a placebo arm to this there. So young people, if they're enrolled in the trial and they move forward, they will get the active gene therapy. So there's no placebo that's being given, which again I think is very appropriate for this stage of study there. Obviously then there's the treatment and dosing. So it's a one-off dose. As we said, it's going to be delivered via the intrathecal injection there. And then afterwards there's a long period of follow-up where there is a lot of tests that are done there. There's a lot of assessments. Again, it's all around that focus on the safety, making sure that it is appropriate to continue with the study and to expand to other young people, and getting that glimmer of the efficacy data to inform the subsequent trial designs there. So, the regulators are all over this and very approved there. So the pathway, once human clinical trials have begun, is kind of outlined here. So the starting point is the trial sponsor, so Taysha, collecting all of the data from the animal models, from the laboratory there, putting together a portfolio to present to the regulators, so in the UK that's the MHRA, to say, look, we've got this data to show that we think that there's a chance that this intervention will work, and we think the safety data from that there. That's all submitted to the regulators, and the voice of the patient advocate, as Chelsea's explained there, and that's passed the hurdle in the UK. So the MHRA has approved the trial to open in the United Kingdom. Clinical data then is obviously collected throughout the clinical trial, and there are certain time points during the study, certainly early on in those phases after each of the dosing, where the independent data monitoring committee decides whether things will move forward, and there's interim reports that are often presented to the regulators to allow things to move forward. At the end, when all the data is collected, that's then presented to the regulators, and then they make the decision as to whether the medication works and is safe enough to be licensed. The extra thing in the UK, which again is something for the future to discuss, is then the commissioning. So the MHRA decides a drug works and it's safe. NICE then has to decide whether the NHS is going to fund that. But that's a step which data is conducted in parallel. So, talking about this study specifically, the REVEAL studies. So there are two arms to this there. So these are both phase 1-2 studies, but we have the adolescent and adult study. So this is running currently in Canada, with sites to open in the United States. It's enrolling females with Rett Syndrome from ages 12 and older. So when it originally started, it was a higher age group, but that's then been brought down. Taysha has dosed 2 participants at the low trial level, and I think what's very exciting that I've only found about myself in the last few days is that there's plans to dose a 3rd participant in the 2nd quarter of this year, but it's going to be at the higher dose level. The paediatric study is running with a site open in the United States there. That's for children from 5 to 8 years of age there, and 2 participants have been dosed so far at the lower level there. This is obviously a tremendous sort of advance as a community. We have this trial open and this data that's beginning to be collected. Am I talking to this one or are you, Chelsea? I'll go for it. Okay, that's fine then. Okay. So in the UK, the plan is that we aim to open as a centre later this year. There are always hiccups. We had hoped that we would be in a position to open earlier on, but unfortunately there's been a few setbacks that we're getting back there. The Medicines Health Regulatory Authority approved the trial to open in the United Kingdom back in January, so that approval in place there, that hurdle's been passed there. We're not actively enrolling just at the moment, but of course once we are open as a clinical site, Reverse Rett will be acting as the advocacy group and will be helping us share information and coordinate the enrollment of the trial there. There is data and information which is on the clinicaltrials.gov website. I mean, we've shared all of it today, but there is a resource that people can tap there. And as we said before, the best way that families can register their interest is with the registry. And obviously, once we are open for clinical recruitment, we'll be reaching out to potentially eligible families. So the timeline, as we said, I think we've covered most of this actually, but we have quite a lot to do in each of the clinical trial sites where things have been a little bit slower than we might have liked there. So we obviously have to have the training appropriate. There's all of the internal processes in terms of the different regulatory processes within the hospital there. And as I say, because this is such a complicated intervention with a lot of monitoring that needs to be in place afterwards, there's just really a lot of moving parts to get lined up before we're safe to open up there. And the very worst thing we could possibly do is open, dose a young person, and then not have everything in place to conduct the rest of the trial. Data is so precious. It's so important that we collect everything, primarily for the safety, but also for the efficacy there. So I think on behalf of myself and Taysha, we just want to really thank Reverse Rett for all of their support for getting the study to this point there. We'll get another round of applause for Rachel and the team there for putting the day together, if that's okay. And I think we're going to move anyways very soon to the kind of Q&A panel, but I'm sure Chelsea won't mind if I say that her and I are very happy to be approached during the break for any questions that people have.